Hello my students, welcome to this video. In this video, I will discuss about physiological effects of auxin. The important physiological effects of auxin are as cell elongation, apical dominance, root initiation, prevention of abscission, parthenocarpy, respiration, and callus formation. Cell elongation. The primary physiological effect of auxin in plant is to stimulate the elongation of cell in shoot. A very common example of this can be observed in phototropic curvature, where the unilateral source of light unequally distributes the auxin in stem tips. In this picture, you can see that the auxin concentration is high at the shaded side in comparison to the illuminated. The higher concentration of the auxin on the shaded side promote the cells to elongate. जिस साइड पर concentration ज़्यादा होगा auxin का, उधर की जो सेल होंगी वो तेजी से elongate करेंगी, जिसकी वजह से जो शूट टिप है वो source of light की तरफ bend होगी, and the response of the auxin begins within 10 minutes after the application of the auxin. Apical dominance. Auxin transport induces apical dominance. In many vascular plants, especially which are tall and branched, if the terminal bud is remain intact, the growth of the lateral bud suppressed. यानी कि अगर जिन प्लांट में एपिकल बड प्रेजेंट रहती है, उन प्लांट्स की लेटरल बड की जो ग्रोथ होती है वो सप्रेस्ड रहती है या रुक जाती है. If we remove this apical bud, the growth of the lateral bud can be done. अगर हम इस एपिकल बड को रिमूव कर दें तो जो लेटरल बड्स होती हैं, लेटरल ब्रांचेस होती हैं, उनकी ग्रोथ इजीली हो जाती है. तो this phenomenon in which the apical bud dominates over the lateral bud and does not allow the lateral buds to grow is called the apical dominance. स्कूक एंड थाइमन ने एक एक्सपेरिमेंट किया जिसमें उन्होंने इंडोल एसिटिक एसिड को अगार ब्लॉक में लिया और बीन प्लांट की टिप को कट करने के बाद उस अगार ब्लॉक को ऑक्सिन के साथ प्लेस किया. He applied the auxin to decapitated stem, and he found that it suppresses the lateral buds. If the agar block is placed again to the decapitated bud but without auxin, the growth of the lateral buds takes place. It means auxin जो होता है वो lateral bud की ग्रोथ को रोकने के लिए रिस्पॉन्सिबल होता है. Here another plant hormone, cytokinin, also work, which act as the inhibitor of the auxin transport and to overcome the apical dominance. Root initiation. The higher concentration of the auxin initiates more lateral branches in roots. But in contrast to the stem, the high concentration of the auxin inhibits the growth of the roots. It means stem में higher concentration जो होती है वो उसकी growth को promote करती है, लेकिन root में higher concentration जो होती है वो उसकी growth को suppress करती है या inhibit करती है. लेकिन higher concentration of the auxin is always initiate the growth of lateral roots. But for this, the indole butyric acid and naphthalene acetic acid are used by the plant growers because these synthetic auxins are more effective in new root initiation, and these roots are not easily destroyed and persist for the longer time. Here in this picture we can see how indole butyric acid promotes the growth of little buds. Prevention of abscission. Natural auxins have controlling influence on the abscission of leaves, fruits and flowers. What is the abscission? Shedding of the leaves, flowers and fruits is known as the abscission. And abscission always takes place due to the formation of a distinct zone which is known as the abscission zone. Abscission zone is formed between petiole or pedicel or fruit stalk and stem at the point of attachment. In this picture, we can see that concentration of the auxin is high at the proximal end, and in this concentration of the auxin is high at the distal end. It means if the auxin concentration is high at the proximal end which is near to the axis or point of the origin या point of the attachment. अगर इस पॉइंट पर proximal end पर ऑक्सिन की concentration हाई होती है तो abscission हो जाता है, लेकिन distal end पर यानी कि वो end जो मेन एक्सिस से दूर है वहां पर ऑक्सिन की concentration हाई होती है तो abscission नहीं होता है. Due to the higher concentration of the auxin at the proximal end, the ethylene formation also promoted. It means अगर proximal end पर ऑक्सिन की concentration हाई होती है तो ये इथाइलिन की ग्रोथ को प्रमोट करती है, और इथाइलिन जो हार्मोन होता है वो एब्सिशन जोन के फॉर्मेशन में हेल्प करता है. अगर डिस्टल एंड पर ऑक्सिन की concentration हाई होगी तो इथाइलिन की फॉर्मेशन नहीं होगी और एब्सिशन जोन का फॉर्मेशन भी नहीं होगा. तो by maintaining the high concentration of the auxin at the distal ends, abscission can be stopped. Parthenocarpy. The formation of the seedless fruits without fertilization is known as the parthenocarpy. And auxin-induced parthenocarpy. If external application of the auxin is applied on flowers, seedless fruit develop in tomato, apples, oranges, cucumber, etc. And it has been found that in parthenocarpic ovaries, the concentration of the auxin was high than in ovaries of the plants which produce fruits only after fertilization. Parthenocarpic fruits economically very useful as they are used to make jam, jellies, pickles and squash, etc. Respiration. Auxin stimulates respiration. Auxin induces synthesis of various enzymes, and by induction of the synthesis of enzymes, the rate of the respiration can be stimulated. Increasing activity of enzyme enhance the respiration. And the auxin may increase the rate of respiration indirectly through increased supply of ADP यानी कि adenosine diphosphate by rapidly utilizing the adenosine triphosphate in the expanding cells. Callus formation. What is the callus? An undifferentiated mass of the cells is known as the callus. And if the growth of the callus is not normal, auxin can be applied for the proper growth. So auxin play very important role, very active role in the cell division, and by this the callus growth can be increased. Thank you very much. If you like my video, please like, comment and share.